Hi everyone, my name is Erin Parlow, and today I'm going to be discussing my honors capstone project, kind of what I learned and what the focus of the project was this semester. My honors capstone advisor was Dr. Geeta Mehta, and you can see the title of my project on this slide. So before I get started, I'd like to give a little bit of background about myself. I am a senior studying materials science and engineering. I'll be graduating at the end of this semester in 2022. I have minors in Spanish and biochemistry, and obviously I am an engineering honors student. My capstone advisor, as I mentioned previously, was Dr. Geeta Mehta, and all of the work that I did for this project was done through the Engineered Cellular Microenvironments Lab, where I have been working as an undergraduate research assistant since I was a sophomore. The scope of the research of this lab is to understand ovarian cancer in kind of all of the stages of the disease and also potential treatment options for the disease as well. And the subset of kind of this research area that my group has been focused on is understanding the mechanical response to shear stress that ovarian cancer cells face and how it promotes their metastasis into more severe disease states. I want to start by giving a little bit of background about ovarian cancer itself. It is one of the most deadly forms of cancer experienced, the fifth most deadly form of cancer for women, and is the most deadly form of cancer for the female reproductive tract. It has poor treatment outcomes and low survival rates, and that's mostly due to a lack of early diagnostic methods, which makes treatment difficult once a person is diagnosed with ovarian cancer. And so you can see from these statistics how broad the impact is for understanding and being able to treat the disease better. So as I mentioned, my subgroup of the lab is interested in understanding how shear stress promotes metastasis of ovarian cancer. Previous work has shown from our lab that shear stress is one of the factors that promotes metastasis of ovarian cancer cells. And so now we're trying to understand from a biochemical perspective why that occurs. And so previous research has shown that in renal cancers, the surface marker MUC15, which is a glycoprotein composed of a protein domain and carbohydrate domains that are extracellular, increased expression of this surface marker is correlated with promotion of the PI3K AKT pathway, which results in phosphorylation of AKT, and then promotes migration, invasion, and metastasis of ovarian cancer. And so our lab is hoping to show that this same pattern is present in ovarian cancer as well. So previously, we've done work with RNA-seq experiments, which has shown that MUC15 is one of 50 underexpressed genes in ovarian cancer, which means that when MUC15 is not expressed, it allows for the promotion of that signaling cascade. And so this allows for the metastasis. And so to kind of tease apart how MUC15 expression patterns relate to the metastasis, we've been looking at MUC15 protein quantification experiments as well. And we want to see if MUC15 expression trends follow expectations. And so that's kind of a project that's going on right now. And part of that is discovering optimal Western blotting parameters, because Western blot is kind of the preeminent experiment method to quantify different proteins of interest in cells. And so my project specifically was to look at PAKT expression in cells with varying levels of MUC15 expression. And so we had 15 genetically engineered cell lines. We had a control, just a basic ovarian cancer cell to give us baseline MUC15 expression in cancer cells. We have up-scrambled and down-scrambled genetic controls to show us that the genetic engineering has like no effect on the expression patterns of MUC15. And then we have an upregulated MUC15 cell line and a downregulated MUC15 cell line. So through these cell lines, we're hoping to show that in the upregulated MUC15 cell line, there is a lesser amount of PAKT than the control. And then the downregulated MUC15 cell line, there's a higher amount of PAKT expression than the control. And this is because that previous renal cancer cell paper showed us that when MUC15 is downregulated, PAKT is upregulated, and this causes cancer metastasis. And so I conducted Western blots on cell lysates from all of these cell lines to try to make that connection. So here are the results. So as I kind of mentioned previously, our lab has been working on developing optimal Western blotting parameters in order to undergo these experiments that we are proposing in order to connect MUC15 expression with this PI3K AKT signaling cascade. And this kind of just requires a lot of work because Western blotting is a little bit of a finicky experimentation method. So it requires a lot of fine tuning in order to show reliable and predictable data. And so these were issues that I kind of ran into when I started my experimentation this semester on the capstone. So got a lot of blotchiness in the samples, unresolved data, and unclear separations. And so these were all challenges that I had to work through and actually spent a better part of the semester fine-tuning parameters in order to get resolved data. And so I got data sets that look like this, which shows us clearly the expression of the different proteins of interest. But as I will discuss later, we didn't really get conclusive results about the expression patterns. But what you can see in this data is that the beta-actin protein, which is a protein that just indicates whether or not the cells are alive and proliferating as they should, is constant across all the cell lines. And that's kind of what we expect. Beta-actin is a form of a control. But the PAKT is also constant across cell lines, and this is not what we would expect. As I mentioned previously, we'd expect lower amounts of PAKT in the VAR2 cell line, which has increased MUC15 expression, and higher amounts of PAKT in the 696 cell line, because this has lower amounts of MUC15 expression. And then you'd expect the reverse trend in the AKT cell lines, as you see in the PAKT cell lines. But for both AKT and PAKT, it's about constant expression across all of the cell lines. And so to get into the conclusions and next steps, so based off of all of that fine-tuning of Western blotting, I was able to determine the parameters of greater than 40 milligrams of protein and overnight incubations as the best for showing expression. But the housekeeping genes were consistent across cell lines, so the cells are growing and proliferating as we'd expect. However, there are no noticeable differences in expression of PAKT and AKT across cell lines, and there's a lot of reasons why this might occur. One of those is that we experienced a lot of contamination issues in the lab this semester, and this could have been affecting the expression patterns and the results that we saw. And so based on these understandings, the next steps that I'd recommend for this project are starting with new cell lines that haven't experienced a contamination and trying to complete repeatable resolved Western blot analysis with these protein cell lines, and then compare the expression patterns with the expected MUC15 expression patterns. And then kind of after that, there's two additional types of experimentation that we can undergo in order to connect all of this microbiology work back to the physical work that we've previously done in the lab with shear stress. And so ELISA and co-immunoprecipitation columns are two ways to determine how that physical signal is being translated to a chemical signal, whether it's to the formation of protein dimers or whether or not it's losing the glycoproteins to the external environment when shear stress is detected. So I have a few questions about my project that I'd like to answer for the honors community. The first is how I got involved in this project. And as I mentioned previously, I've been an undergraduate research assistant since I was a sophomore. And this is also what helped me with my capstone, getting connected to my capstone advisor. I'm really passionate about the research that our lab does, and I was really excited to have my own personal project to work on this semester. And then the challenges that I encountered were, again, just not having reproducible data because of the finickiness of the Western blotting and microbiology research. And so that I would just say is really important to know that things might take a while, but if you keep working at them and kind of refining your methods and keeping good accounts of what you're doing, it is able to learn something that can help projects in the future. I'd like to thank Dr. Gita Mehta and Eric Horst for supporting me in this project and through the work on this capstone. And I'd also like to thank the honors program for providing community and guidance for the completion of this project. I really enjoyed my time in honors, and this project has really been a great experience to add on to my senior year. That's all I have for now, but thank you for listening to my honors presentation.